Welcome back to the deep dive. It's really good to be back. Today we are stepping away from the frantic, fast-paced world of viral outbreaks and, leaving behind the rapid binary fission of bacteria. We're entering a kingdom that is, well, it's quieter, it's slower, often overlooked, but in many ways, much more sinister. We're talking about the kingdom fungi.
Yeah, it's a kingdom that often gets treated as a footnote, doesn't it? I mean, in medical school, you spend months on bacteria, weeks on viruses, and then maybe, 2 days on fungi? It feels like an afterthought. But in the actual clinical world, especially in the modern hospital where we have patients living longer with severely compromised immune systems, fungi are the ones that keep infectious disease doctors awake at night.
Right, and that is exactly why we are focusing on Murray's Medical Microbiology, Chapter 60 today, along with some supplementary diagnostic principles from gelates. We aren't just looking at what fungi are biologically, we're diving into the detective work, the laboratory diagnosis. Because as the source material makes painfully clear, diagnosing a fungal infection is not at all like diagnosing strep throat. It requires a completely different mindset.
Absolutely. With bacteria, you have this luxury of speed and numbers. You swab a throat, you plate it, and you have millions of colonies the next morning. It's like playing checkers. It's fast, it's reactive. Fungal diagnosis, that is 3D chess. You're dealing with organisms that hide deep in the tissue, organisms that mimic human cells, and organisms that grow so slowly that, honestly, the patient might be dead before the colony ever appears on the plate.
Which is a terrifying thought, but it really sets the stakes for this discussion. The mission for this deep dive is to decode the diagnostic toolkit for you. We need to build a mental model of how to visualize, grow, and detect these organisms before they checkmate the patient. Murray breaks this down into a really logical roadmap, and I want to follow that structure closely today because it mirrors the actual workflow of a clinical lab.
It does. It moves chronologically, which is really the only way to understand the process.
Yeah. So we start with the patient at the bedside clinical recognition, then we move to the physical sample specimen collection, then the rapid tests, direct microscopy, then the gold standard culture, and finally we'll hit the modern era of non-culture methods like antigens and molecular markers.
It's the journey from the bedside to the bench and back again. And every single step has major pitfalls.
Let's start right at the beginning then. Section 1, clinical recognition. Murray introduces this concept of the index of suspicion. It feels like a slightly vague term at first glance, but in mycology, it seems to be the single most important diagnostic tool. Why is that the starting line?
Because with fungi, you cannot find what you aren't looking for. That is the golden rule. Bacteria are often loud, right? They cause high fevers, skyrocketing white blood cell counts, massive rapid shifts in blood pressure. They announce themselves. Fungi are often the silent killers, specifically because they tend to strike when the host is already weak. So if you have a patient who is deteriorating and you've thrown every broad-spectrum antibiotic in the bucket, them vancomycin, meropenem, zosyn, and they just aren't getting better, your brain has to pivot. You have to actively think, could this be fungal?
It's that pivot that seems to be the hardest part for a new clinician to master. Yeah. You get tunnel vision on bacteria. Murray outlines specific patient profiles, almost like archetypes, that should trigger this pivot immediately. I want to walk through these because they feel like high-yield exam scenarios for you listening, but also critical life-saving patterns to recognize on the wards.
They're the classic board exam questions, yes, but they represent real human suffering. Recognizing these patterns early is often the literal difference between life and death.
Let's take the first one, the neutropenic patient. Someone with a dangerously low white blood cell count, usually neutrophils, often from chemotherapy for leukemia or lymphoma.
Right. Neutrophils are your primary first line of defense against molds. They are the soldiers on the wall. In a neutropenic patient, that wall is totally unmanned. If this patient spikes a fever, and especially if they start developing skin lesions, you need to be terrified of Aspergillus or Fusarium.
Fusarium specifically has a very distinct, almost pathognomonic presentation in these patients, doesn't it? Murray highlights this.
It does. It causes these necrotic black skin lesions. We call them eczema gangrenosum-like lesions. It looks like the tissue is just dying and turning black right before your eyes, and the mechanism behind that is terrifying. Fusarium is angio-invasive. That means the fungus actually invades the blood vessels themselves. It clogs them up, cuts off the blood supply to the skin, and the skin infarcts and dies. If you see that in a leukemia patient, you don't wait for a culture. You assume it's a mold, and you start aggressive antifungals immediately.
Okay, so neutropenia plus skin lesions equals Aspergillus or Fusarium. Let's look at the second profile, the diabetic patient, specifically the patient in diabetic ketoacidosis, or DKA.
This one is the stuff of nightmares. DKA creates a very specific biochemical environment in the body. The blood becomes acidic, that's the acidosis, and there is extremely high blood sugar. Plus, the acidic environment frees up iron in the blood. There's a group of fungi called the mucormycetes. You'll hear them called zygomycetes too. The taxonomy shifts around, but the clinical picture is the same. Specifically, the genus Rhizopus. They absolutely thrive in this exact environment.
Why that specific environment, though? What's the mechanism?
Rhizopus has an enzyme system, ketoreductase, that lets it utilize the ketone bodies produced in DKA. It essentially allows them to thrive in acidic, high glucose, high iron conditions. It's like rocket fuel for them. And where do they usually enter?
They go for the sinuses.
The start of the sinuses. But they don't just stay in the nasal cavity like a common cold. They are incredibly aggressive. They erode straight through the bone, into the orbit of the eye, and eventually directly into the brain.
Murray mentions a specific physical sign to look for here, the black Escher.
Yes. It's a black, dead patch of necrotic tissue, usually found on the roof of the mouth, the hard palate, or inside the nasal turbinates during an exam. If you see a diabetic patient with severe sinus pain, facial swelling, and a black spot inside their mouth or nose, that is a surgical emergency. That is mucormycosis until proven otherwise.
It's interesting you say surgical emergency, not just medical.
You can't just treat that with IV drugs. Just like Fusarium, Rhizopus invades the blood vessels and kills the tissue. Dead tissue has no blood supply. If there is no blood supply, the systemic antifungal drugs physically cannot get to the fungus. You have to go in and cut the fungus out. The surgeons have to debride it until they hit healthy bleeding tissue.
That is a very visceral image, but it really underscores the urgency.
Let's move to the third major group, the HIV/AIDS population.
Here, your index of suspicion shifts again. In patients with profoundly low CD4 counts, we aren't looking for Aspergillus or Mucor as much as we are looking for the pathogenic yeasts and dimorphics. Cryptococcus neoformans is the big one, causing subacute meningitis, Histoplasma capsulatum causing disseminated body-wide disease, and of course Pneumocystis jirovecii causing a very specific, progressive type of pneumonia.
So the clinical picture effectively acts as your first diagnostic filter. It narrows the suspect list before you even walk the sample down to the lab. Exactly. You walk into the lab with a working hypothesis. You say, I am looking for Cryptococcus, or I am looking for Mucor. That hypothesis guides literally everything that happens next.
And what happens next is getting the sample. This brings us to specimen collection. And reading Murray, it seems like there are two major ways to completely mess this up before you even get to the bench. The first is contamination.
This is a massive headache in clinical mycology. You have to remember, fungi are ubiquitous. They are in the air we are breathing right now in this room. They are on your skin. They are on the hospital tray. If you leave a piece of bread out, mold grows on it. That mold came from the air.
So if you are sloppy
with your collection, say you take a swab and you accidentally brush it against the patient's cheek or tongue on the way out of the throat, you're going to pick up environmental molds or harmless commensal yeasts.
And then the lab grows a mold and the doctor thinks, I found the killer.
But really, they just found a hitchhiker. Distinguishing between a true pathogen and an environmental contaminant is the hardest part of reading fungal cultures. So, aseptic technique is rule number 1. You have to meticulously clean the site, use sterile containers, and minimize any exposure to the room air.
Rule number 2 seems to be about quantity. The volume rule. Murray is very strict about this, and Jowett backs this up regarding diagnostic principles.
Yes. This is a common frustration for microbiologists. With bacteria, they replicate so incredibly fast, doubling every 20 minutes, that you can often get away with a tiny swab. You drop it in broth, and boom, you have plenty of bugs. Fungi are completely different. They are often present in much lower numbers in the tissue.
So a standard dry swab just isn't going to cut it.
A dry swab is useless. If you send a dry swab with barely anything on it to the mycology lab, they're going to report no growth. Not because the fungus wasn't there, but because you didn't give them enough of it to recover. You need a chunk of tissue. You need a full vial of fluid. You need deep scrapings. You need volume.
Now, speaking of tissue biopsies, there is a specific warning in Murray that I found absolutely fascinating. It's what I'm calling the grinding trad. This feels like a detail that really separates the novices from the experts.
It absolutely does. And it's a mistake that happens in real hospitals, often leading to tragic outcomes. It's one of those things where knowing the basic structural biology directly impacts a critical lab procedure.
So paint the scenario for us. We have that diabetic patient with the sinus infection. We suspect rhizopus. We take a biopsy of the sinus tissue and send it to the lab. What happens next in the standard default protocol?
The standard protocol for processing a piece of tissue in a microbiology lab is to homogenize it. You put the tissue in a sterile tissue grinder, think of a mortar and pestle or a small mechanical blender, to break up the human cells. This releases the bacteria or fungi trapped inside so you can spread them evenly on the agar plates.
That sounds completely logical. Why is that a problem for rhizopus?
It's a problem because of the unique structural anatomy of the mucormycetes. We have to visualize the microscopic structure here. Most molds, like Aspergillus, have hyphal. Those are the long filaments that have cross walls called septa. These cross walls add structural support, kind of like the bulkheads in a submarine.
Okay, so they are compartmentalized.
Right. But mucormycetes, they are described as pauciseptate or non-septate. They are basically long, continuous, open tubes. There are almost no cross walls.
Like a long, fragile glass straw filled with fluid.
Exactly. A liquid-filled glass straw. Now imagine putting a glass straw into a mechanical tissue grinder. What happens?
It completely shatters.
It shatters. The mechanical force shears the straw. Because there are no cross walls to seal off the damage, the cytoplasm leaks out, all the nuclei spill out, and the organism dies instantly.
So you grind the tissue, you plate it, and nothing grows.
Nothing grows. The lab officially reports no fungal growth. The clinician reads that and thinks, oh good, it's not mucor. Maybe it's a weird bacterial infection. They stop the empirical antifungals, and the patient dies from an untreated fungal invasion.
That is chilling. So the physical act of processing the specimen can literally kill the diagnosis if you don't understand the underlying biology.
Correct. This is exactly why clinical communication is key. If you suspect mucor, you have to write it clearly on the lab requisition form. Rule out mucor. That specific note tells the lab tech, step away from the grinder. Instead, they will carefully mince the tissue, chop it into small pieces with a scalpel to preserve those fragile non-septate hyphae.
That is a massive life-saving distinction. Mince, don't grind.
Mince, don't grind. It shows up on exams, but more importantly, it saves lives.
Okay, so we have our specimen. We've collected it safely. We haven't ground it to dust. Now we need answers. We know culture takes time. We'll get to that. But the patient is sick right now. This brings us to section 2, direct microscopy.
This is the speed run. Direct microscopy is really the only way you get a fungal diagnosis in under an hour. It allows you to start targeted treatment before the sun goes down.
Murray lists a whole toolkit of stains here. I want to go through these, but I want to focus on the why for you listening. Why do we need so many different ways to look at a slide? Let's start with the most basic one. KOH, potassium hydroxide. I've heard this called the clearing agent.
That's the perfect name for it. Think about it. Imagine you scrape some skin off a patient with a severe rash, or you take a thick nail clipping. If you put that sample directly under a microscope, what do you actually see?
Just a wall of skin cells.
Layers and layers of dead skin cells. Keratin, protein, cellular debris, oils. It's an absolute mess. It looks like a dense pile of leaves. You cannot see the microscopic fungus hiding in there because the massive human cells are completely blocking the view.
So KOH acts as a chemical cleaner.
Yes. Potassium hydroxide is a strong alkali. When you mix it with the sample on the slide, it starts to digest the protein. It dissolves the human skin cells. It breaks down the tough keratin. But, and this is the vital key, it does not dissolve the fungus.
Why not? Why is the fungus immune to this strong base?
Because fungal cell walls aren't made of keratin or standard proteins. They are made of chitin. That's the exact same tough, rigid material that insect exoskeletons and crab shells are made of. Chitin is incredibly chemically resistant. It easily resists the alkali. So after the KOH does its work, the human cells dissolve into a clear soup, and the fungal hyphae are left behind completely intact and visible.
It clears the fog so you can actually see the enemy. But Murray mentions a practical tip here. It takes time.
It does take time. If you look immediately after adding the drops, it might not be clear yet. Sometimes you have to warm the slide gently over a flame or a hot plate to speed up the chemical reaction, but you have to be very careful not to boil it or you'll crystallize the KOH and completely ruin the view. It's a bit of an art form in the lab.
Now, KOH is great, but it requires a really trained eye to spot those transparent hyaline hyphae against a clear background. It's like looking for glass in water. There is a way to make them pop out, though. Calcofluor white. I think of this as the highlighter.
That is exactly what it is. Calcofluor white is a fluorescent stain. It has a very high affinity for chitin and cellulose. It binds specifically to the fungal cell wall,
And then you turn on the UV light.
and the fungus just lights up. Depending on the specific filter you use on the fluorescent microscope, it glows a brilliant blue-white or sometimes a bright apple green. It is virtually impossible to miss.
So the main advantage here is the speed of screening the slide.
Massive advantage. With a standard KOH prep, you have to scan the slide very slowly, usually on high power, looking for faint ghost-like lines. With Calcofluor, you can scan the slide at low power very quickly. If there is a fungus anywhere on that slide, it will scream at you from the dark background. It's the difference between searching for a flashlight in a dark room versus searching for a clear glass straw in a swimming pool.
Okay, so KOH clears the junk, Calcofluor makes it glow. Now, what about the gram stain? I always associate gram staining strictly with bacteria. Purple for positive, pink for negative. Does it actually work on fungi?
It's the absolute bread and butter of the microlab, so literally every specimen gets gram-stained eventually. And yes, it does work, but you have to know what you're looking for. Fungi are generally considered gram-positive. They hold the crystal violet dye.
So they look purple.
They look purple. Candida species, for instance, appear as large purple budding ovals. I want to really emphasize large here. A yeast cell is huge compared to a typical bacterium. A Staphylococcus bacteria is a tiny dot. Candida is a giant oval. If you're looking at a blood culture smear and you see giant purple footballs, that's Candida.
But Murray mentions a caveat here. They aren't always pretty purple ovals.
True. Sometimes, especially with older cells or different species like Cryptococcus, they just don't hold the stain well. They can look speckled or gram variable, or if the fungal cell wall is damaged, they might even look pink. But as a general rule, if you are thinking fungi on a gram stain, think purple.
Next up is the Giemsa stain. This one seems to have a very specific, almost niche application. I call this the intracellular hunter.
Very specific. Giemsa doesn't stain the outer fungal cell wall particularly well. It stains the internal structures, the cytoplasm, and the nucleus. It is particularly good at showing you things that are hiding inside the host's own cells.
And there's one classic association here that every medical student memorizes for the boards.
Histoplasma capsulatum.
Why histoplasma specifically?
Because histoplasma is a facultative intracellular pathogen. It actively lives and replicates inside our macrophages, the very immune cells that are supposed to eat and destroy invaders. If you have a patient with disseminated histoplasmosis and you take a bone marrow aspirate or a peripheral blood smear, you stain it with Giemsa. You aren't looking for free-floating mold, you are looking for a macrophage, a big host cell that is absolutely stuffed full of tiny blue-purple oval yeasts.
The swarm inside the cell.
Exactly. It looks like the macrophage has the measles. It's also useful for identifying pneumocystis cysts in respiratory samples, but the histoplasma macrophage connection is the one that shows up on board exams constantly.
Finally, for microscopy, we have the heavy hitters from the pathology lab. The tissue stains. If a biopsy goes to surgical pathology instead of micro, they don't usually do gram stains. They do H&E, hematoxylin, and eosin. But Murray explicitly says H&E isn't enough for fungi.
H&E is the standard pink and purple stain for all human tissue. It shows the architecture of the human cells beautifully, but fungi can easily blend right in. Their cytoplasm stains pink, just like human cytoplasm. If there are only a few hyphals scattered in the tissue, they can be completely invisible on H&E. That's why if you even suspect fungus, the pathologist needs to order special stains, specifically GMS and PAS.
Let's break those down. GMS, gamorreum methenamine silver. We call this the silver standard.
It really is. The chemistry here involves silver nitrate. The silver actually precipitates directly onto the polysaccharides in the fungal cell wall and turns it black, pitch black. And they usually counterstain the background tissue a light pale green, so you get these incredibly crisp, black, high contrast outlines of the fungus against the soft green background.
High contrast really seems to be the theme here for finding them.
Maximum contrast. Even if the fungus is dead or dying from prior antifungal therapy, the silver will still stain the residual wall. This is the absolute best stain for seeing the morphology in tissue. Is it branching at 45 degrees? Is it septate? GMS gives you the sharp silhouette you need to answer those structural questions.
And PAS, periodic acid shift.
PAS stains the complex polysaccharides, the sugars in the fungal wall, a bright pink or magenta. It's also very good, vastly superior to H&E for this purpose. It really helps you see the fungus stand out against the background tissue. But most mycologists and pathologists would argue that GMS is slightly better for initial screening simply because that black-on-green contrast is so sharp and unforgiving.
So to recap the speed run of microscopy for everyone listening, KOH to clear the junk, Calcofluor to make it glow in the dark under UV, Gram stain shows us the giant purple yeasts, GMSA finds the histo hiding inside the macrophages, and GMS paints the walls black for the pathologist.
You've got the toolkit, and remember, all this happens in the first few hours after the sample arrives.
All right, we've seen the enemy. Now we need to capture it and interrogate it. Section 3. Culture. The growth engine.
Culture is still the absolute gold standard. Despite all our modern molecular advances, honestly, nothing beats growing the live organism. It allows you to test for specific antifungal drug resistance, and it allows for definitive morphological identification. But it requires patience.
And it requires the right soil. We're back to our gardening metaphor. You can't just throw seeds on concrete and expect a crop. You need the right media. Murray categorizes these into non-selective and selective media. Let's start with the non-selective, the buffet.
Right. This is usually something like brain heart infusion, agar, BHI, or SABI, which is sabourode plus BHI. These are extremely rich, nutrient-dense agars. They often have sheep blood added to them. Non-selective means exactly what it sounds like. We aren't trying to stop anything from growing. We want to catch absolutely everything in that sample.
But if everything grows, doesn't that include all the normal bacteria?
Yes. And that is a huge problem. If you have a sterile specimen like spinal fluid or deep tissue, non-selective media is great. But if you have a sputum sample from the lungs or a skin scraping, those are naturally full of normal bacterial flora. Bacteria replicate every 20 minutes. Fungi might take 20 hours to double. If you put them on the exact same plate, the bacteria will swarm the entire plate and eat all the nutrients before the fungus even wakes up.
So you need to clear the field. Enter the weed killer's selective media.
Exactly. We need to artificially suppress the competition. Step 1 is adding antibiotics to the agar. Penicillin, streptomycin, gentamicin, chloramphenicol. These effectively kill off the bacteria.
That gives the fungi a fighting chance. But Murray mentions another layer of selection, cyclohexamide. This seems to be a very, very important chemical for students to understand.
It is critical. Cyclohexamide is actually an antifungal agent itself. It targets eukaryotic protein synthesis.
Wait, we intentionally put an antifungal in the fungal culture media. That seems completely counterintuitive.
We do. Because just like there are weeds in a vegetable garden, there are weeds in the fungal world. These are the saprophytic molds, the rapid environmental growers that live on bread or in the dirt outside. Penicillium, environmental rhizopus, things like that. They grow incredibly fast and they are extremely fuzzy. If just one spore from the air lands on your patient's plate, it will cover the entire thing in 48 hours, completely ruining your chance to find the slow-growing pathogen underneath.
So cyclohexamide kills the fast-growing environmental weeds. That sounds perfect. Why don't we just use it on every single plate in the lab?
Because of the cyclohexamide trap. This is a massive exam alert and a real clinical pitfall. It turns out some of the most dangerous human pathogens are also sensitive to cyclohexamide.
We are accidentally poisoning our own witness.
Exactly. If you only use media with cyclohexamide, you will kill the very thing you are desperately trying to diagnose.
We really need to know this list, the sensitive list. Who dies in the presence of cyclohexamide?
The big one, the one you absolutely must memorize for the boards, is Cryptococcus neoformans, the major meningitis bug. It is completely inhibited by cyclohexamide.
Okay, so if I suspect crypto in a CSF sample and I use the weed killer media, I get a false negative.
Correct. Other really important ones are some species of Aspergillus, like A. fumigatus sometimes, some Candida species like C. parapsilosis or Creuset, and crucially, once again, the Mucormycetes, Rhizopus, Mucor.
The Mucormycetes again. They are fragile to grinding, and they are fragile to cyclohexamide. They are incredibly hard to catch.
They're very elusive organisms in the lab, so the protocol in a clinical lab is non-negotiable. You must use paired media. You always set up one plate with cyclohexamide to kill the weeds and one plate without cyclohexamide to catch the sensitive pathogens. You need both views to get the full picture of what's in that sample.
Makes perfect sense. Redundancy is safety in the lab. Now, there's one more special media requirement Murray mentions that feels like a piece of trivia until you realize how common the infection actually is. The olive oil clue.
Ah, yes. Malassezia furfur.
Malassezia. This is the fungus that causes tinea versicolor. Those flaky hyper or hypopigmented patches on the skin that people often notice in the summer when they tan.
That's the common outpatient presentation, sure. But in the hospital setting, it can be much more serious. It can cause severe catheter-related bloodstream infections, especially in neonates in the NICU or adult patients receiving TPN, total parenteral nutrition.
And TPN is full of lipid emulsions, fats.
Precisely.
Yeah.
The key biological fact about Malassezia is that it is a lipophilic yeast. It literally loves fat. In fact, it needs long-chain fatty acids to build its cell wall. It cannot synthesize them on its own from basic sugars. It's a metabolic cripple without exogenous fat.
So if you put it on a standard agar plate with just glucose,
It won't grow, it just starves to death.
do you have to manually feed it fat?
Literally. In the lab, if the doctor suspects Malassezia, maybe because the patient is spiking fevers while on lipid infusions, we will streak the blood sample on the plate and then overlay it with a layer of sterile olive oil.
Olive oil, just poured right on top.
Simple, cheap, but incredibly effective. If you come back a few days later and see tiny yeast colonies growing only underneath the oil slick, you have your definitive diagnosis. Malassezia furfur.
I love that, the Italian dressing diagnosis.
Yeah.
Okay, so we have the media sorted. Now, the incubation conditions. Temperature matters in the fungal world a lot more than I initially realized.
It's a key variable. We generally use 2 temperature settings in the incubators.
Yeah.
25 to 30 degrees Celsius. That's essentially room temperature, or slightly above. And 35 to 37 degrees Celsius. That's human body temperature.
And this brings us to what I think is the coolest biological trick in the kingdom, the dimorphic fungi, the shapeshifters.
They are the most fascinating group by far. Histoplasma, blastomyces, coccidioids, sporothrix, paracoccidioids. These organisms have 2 completely different structural forms depending entirely on the ambient temperature.
What is the classic mnemonic for this?
Mold in the cold, yeast in the beast.
Mold in the cold, yeast in the beast. Explain the mechanism behind that shift.
In the environment, out in the soil, in bird droppings, or in the lab incubator at 25 degrees, they grow as a mold. They have branching hyphae. They make tiny spores called microconidia. This is the infectious form. You walk by, kick up the dust, and breathe in the spores. But once those spores land deep in your warm alveoli at 37 degrees, they trigger a massive genetic switch. They completely restructure themselves into a yeast form. They become single-celled, budding organisms.
Why do they do that? Why is yeast better inside us?
This shape is far better for traveling through the bloodstream and evading the immune system. A long branching hypha is easy for the immune system to recognize and attack. A tiny round yeast can hide inside a macrophage like histoplasma does.
So for the lab to officially confirm the diagnosis of a dimorphic fungus, do they just look at the mold on the plate?
Looking isn't enough for definitive proof. You have to prove it can actually shapeshift. You grow it at 25 degrees and you see a mold.
Yeah.
Then you take a sample of that mold, streak it onto a fresh, rich plate, and stick it in the 37-degree incubator. If it converts into a creamy yeast colony after a few days or weeks, you have confirmed beyond a doubt it is a dimorphic true pathogen.
It's literally like catching a werewolf transforming. You have to see both forms to be absolutely sure.
That's a great analogy. And keep in mind, these specific cultures take a lot of time. Yeasts like Candida are fast. They grow in a couple of days. But the dimorphics, histoplasma is notoriously slow. It can take 4 to 12 weeks to grow from a clinical sample.
12 weeks. That's 3 entire months.
Yes. Most labs have a standard protocol to throw away negative fungal cultures after 4 weeks to save space. If you don't explicitly tell the lab, suspect histoplasma, they might toss the plate in the biohazard trash right before the colony finally appears.
Communication is everything. Okay,
Yeah.
let's say we have growth. We have a colony on the plate. Now we need section 4, identification, naming the suspect.
Identification always starts with gross morphology. What does the colony look like to the naked eye when you hold the plate up to the light?
Yeasts versus molds.
Yeasts generally look a lot like bacterial colonies. Pasty, opaque, creamy, smooth. They often smell distinctive, too. Candida smells like baking bread or brewer's yeast. Cryptococcus can look very mucoid or shiny and slimy because of its massive polysaccharide capsule.
And the molds.
Molds are fuzzy, cottony, powdery, filamentous. They have real texture. They physically stand up off the agar plate, and they come in every color of the rainbow: green, black, white, bright yellow. That color almost always comes from the spores they produce on the surface.
But looking at a fuzzball on a plate isn't enough for a medical diagnosis. We need to look closer. Microscopic morphology. This is where we look at the fingerprint of the mold under the scope.
For molds, the identification is almost entirely visual and structural. We look at 2 main things: the hyphae, the actual filaments, and the conidia, the spores and the structures that hold them.
I want to compare the 2 big ones again because this is a constant battle in the differential diagnosis. Aspergillus versus the mucormycetes like Rhizopus. How do you tell them apart under the microscope?
Let's look at Aspergillus first. It's a very highly structured, organized organism. Under the microscope, you see septate hyphae. That means the filaments have those little regular cross walls. They are very uniform and very tidy looking. And when they branch at a characteristic 45-degree angle.
Acute angles. 45 degrees, septate, tidy.
Right. And the fruiting head, but the part that actually makes the spores is iconic. It looks exactly like a dandelion that's gone to seed. You have a straight stalk, the conidiophore, ending in a swollen bulb called the vesicle, with chains of tiny spores radiating out in all directions. If you see a dandelion under the microscope, it's Aspergillus.
Now contrast that with mucor or rhizopus.
Mucormycetes are the messy ones. They have those broad non-septate or pachiseptate hyphae we talked about. They look like twisted, irregular ribbons. The width varies wildly, sometimes fat, sometimes thin, and they branch at very wide angles, often full 90 degrees.
So Aspergillus is tight, acute, segmented. Mucor is broad, open, irregular, wide branching.
Correct. And rhizopus specifically has these root-like structures called rhizoids that actually anchor it down into the agar, hence the name rhizopus meaning root foot.
What about the yeasts? If I have a creamy colony on a plate, how do I know if it's Candida albicans or just some environmental yeast?
Yeasts are definitely harder to tell apart just by looking. Under the scope, they are mostly just little round or oval budding cells. But Candida albicans, which is the most common human pathogen, has a special trick, the germ tube test.
This is a classic lab test. How does it actually work?
It's incredibly simple and elegant. You take a tiny bit of the yeast colony and mix it into a small tube of serum, usually rabbit serum or human plasma. You put it in the incubator at 37 degrees for about 2 or 3 hours. Then you take a drop, put it on a slide, and look at it under the microscope.
And what do you see?
If it is Candida albicans, the yeast cells will have started to sprout. They grow this long, continuous tube-like extension right out of the side of the cell called a germ tube. It looks a bit like a hand mirror or a sprouting seed. There's no constriction where the tube meets the main cell.
And other Candida species don't do this.
Candida tropicalis or Candida glabrata generally do not form true germ tubes in that short time frame. So germ tube positive rapidly equals Candida albicans. It's a cheap, fast, 2-hour way to ID the most common suspect.
But we are in the modern era now.
Yeah.
We don't always rely on sprouting tails and rabbit serum or looking at microscopic dandelions. We have high-tech solutions. Murray talks a lot about MALDI-TOF.
Yes, MALDI-TOF MS matrix-assisted laser desorption ionization time of flight mass spectrometry.
That is a serious mouthful. Let's break down the physics of that simply for the listener.
It sounds like sci-fi, but the core concept is straightforward. Lasers. You take a tiny smear of the colony from the plate, put it on a specialized metal target plate, add a chemical matrix, and put it into the machine. A laser blasts the sample. This vaporizes the proteins inside the fungus and simultaneously gives them an electrical charge. That's the ionization part.
So now you have a floating cloud of charged fungal proteins.
Right. And these charged proteins are accelerated down a long vacuum tube toward a detector at the other end. It's basically a race. The small, light proteins fly very fast. The big, heavy proteins fly slow. The machine precisely measures the time of flight for all these hundreds of proteins to hit the detector.
And that creates a pattern.
It creates a highly specific spectral fingerprint, peaks and valleys based on protein mass. The machine software compares that fingerprint to a massive, continually updated database of known organisms. It matches the pattern and tells you, on screen, this is Candida parapsilosis with 99.9% confidence.
How long does that take compared to old school methods?
The run itself takes minutes. Compared to setting up biochemical sugar assimilation tests that take days to turn color, it is an absolute revolution in turnaround time.
Does it work for everything? Molds and yeasts?
It is amazing for yeasts. Candida, Cryptococcus. It has completely changed how we handle yeast identification. It's getting better for molds, but molds are inherently trickier. Their phenotypes vary more depending on how long they've grown, and their complex chitin cell walls are much harder to break open to get the internal proteins out for the laser. But for common molds like Aspergillus, it's rapidly becoming the standard of care.
So MALDI-TOF is the speed demon of culture ID. But, and this is a big, but even MALDI-TOF requires a colony to grow on a plate first. And as we said, fungal growth can be very slow. Patients die fast. This brings us to the final and perhaps most clinically urgent part of the roadmap. Section 5, non-culture methods.
These are the surrogate markers. We're essentially saying, I can't find the criminal right now, but I can find his fingerprints left at the scene. We are looking for pieces of the fungus antigens, cell wall components, or DNA floating around in the patient's blood or fluid without needing to actually grow the live organism.
Let's talk about antibodies first. Serology, checking the patient's immune response. Is it useful?
Generally, no, it's fraught with issues, and here's why. Antibodies are a record of the past. Fungi are everywhere. Many of us have been exposed to common fungi in the environment at some point. We might have IgG antibodies to Aspergillus or Candida just from living life on Earth. A positive test might just mean you breathed in a spore 5 years ago while gardening. It does not definitively prove you have a life-threatening active infection today.
And there's the major issue of the specific patient population we're dealing with.
Exactly. Think about who gets severe fungal infections. Immunocompromised people, leukemia patients, transplant recipients, advanced HIV patients, people whose immune systems are broken. They physically cannot make antibodies efficiently. So you run a serology test and you get a false negative because their immune system is just too weak to mount a detectable response, even while the fungus is actively killing them.
So serology is mostly out for acute diagnosis. What about antigens? Looking for the smoking gun of the fungus itself.
Antigens are the gold standard for rapid non-culture diagnosis. We are actively detecting shedding parts of the fungal cell wall or capsule in the body fluids. Let's run through the big four because these tests save lives every single day.
Number 1, cryptococcal antigen or CRAG.
This is an absolute superstar test. It specifically detects the glucuronopsilamanin polysaccharide capsule of Cryptococcus neoformans. We used to use the India ink stain to look for the clear capsule halo under a microscope, but India ink is highly insensitive. It literally misses about 50% of cases.
And the CRAG test.
The CRAG lateral flow assay, the LFA, which basically functions and looks a lot like a rapid COVID or pregnancy test dipstick, is incredibly sensitive and specific. You can run it on spinal fluid or serum. If a patient has a headache, altered mental status, and HIV, you run this test immediately. If it's positive, they have cryptococcal meningitis, it is practically diagnostic on the spot.
Number 2, histoplasma antigen.
This one is best detected in urine, though you can do serum too.
Urine. Why urine for a respiratory or systemic fungus?
Because the histoplasma circulating galactomannan antigen is a relatively small molecule, and it gets filtered and highly concentrated by the kidneys. In a patient with severe disseminated histoplasmosis, like an AIDS patient where the fungus is spread everywhere, the urine antigen is extremely sensitive. It's vastly faster than waiting 12 weeks for that bone marrow culture to finally grow.
Number 3, galactomannan. This is the big Aspergillus test.
Right. Galactomannan is a complex carbohydrate molecule found deep in the Aspergillus cell wall. When the mold actively invades human tissue, specifically when it invades the blood vessels, that angio invasion we talked about, it sheds this sugar directly into the bloodstream.
So we test the serum.
We test serum, yes, but we also heavily rely on testing BAL bronchoalveolar lavage fluid. That's the saline fluid washed out of the deep lungs by a pulmonologist during a bronchoscopy.
Why is BAL sometimes better?
Because the lungs are usually where the primary infection is. If you have a neutropenic patient with shadowy nodules on a chest CT, a positive galactomannan, specifically in the lung wash fluid, is a very strong early indicator of invasive pulmonary aspergillosis, often days before it ever spills over into the blood.
And finally, the tricky one, 1-figan-3 beta-D-glucan. We call this the pan-fungal alarm.
Think of beta-D-glucan, or BDG, kind of like a fungal ESR or fungal CRP. It's a general, non-specific marker of fungal trouble. The assay actually uses an enzyme from horseshoe crab blood limulus amoebocyte lysate to detect this specific glucan. And this glucan is a major structural component found in the cell walls of many, many different fungi. Candida, Aspergillus, Pneumocystis.
So if it comes back positive, it tells you, hey, there is a fungus here, but it doesn't tell you which one.
Exactly. It does not identify the genus or species, but it is incredibly useful for its negative predictive value. If the BDG test is solidly negative, it is very, very unlikely the patient has invasive candidiasis or aspergillosis. It gives the clinician the confidence to stop the toxic, expensive empirical antifungals.
But there is a massive blind spot here, a trap that we alluded to earlier with the grinding trap and the cyclohexamide trap.
Yes, there is, and it's deadly if you don't know it. Not all fungi have 1-velar-3 beta-D-glucan in their walls, or at least they don't shed it in detectable amounts. The two big critical exceptions are, you guessed it, the Mucormycetes and Cryptococcus.
So let's play this specific clinical scenario out. You have that DKA patient in the ER with a black sinus lesion and eye swelling. You order a beta-D-glucan blood test because you want to be thorough and rule out fungus. It comes back negative.
And if you don't know the biology, you might falsely think, oh, phew, it's not fungal. Wrong. Mucor is practically invisible to the BDG test. You could completely dismiss a fatal diagnosis because you trusted a lab test that has a known structural blind spot.
That is such a critical point for everyone listening. BDG is blind to mucor and crypto. You have to deeply understand the limitations of your diagnostic tools.
Absolutely. A test is only as good as the clinician interpreting it.
Finally, just a brief word on molecular methods. PCR. We use PCR for literally everything else in micro-COVID, flu, MRSA. Why is it so tricky and less standard in clinical mycology?
Primarily because fungal DNA is literally everywhere. If I ran a high-sensitivity, broad-range fungal PCR on the air in this recording studio right now, I'd find Aspergillus DNA. I'd find Pneumocystis DNA. The risk of environmental contamination and false positives from harmless colonizers is extremely high. We are getting better at targeted PCRs, but it's not nearly as plug-and-play as it is for viruses.
But there is one cool piece of tech Marie mentions here at the end. T2 Candida.
This is genuinely fascinating. It uses T2 magnetic resonance, essentially miniaturized MRI technology. It detects Candida cells directly inside a whole blood sample. You do not need to culture them or extract them first.
No culture at all.
No culture. It takes 3 to 5 hours, not 2 to 5 days. It's extremely fast and can detect incredibly low numbers of yeast cells, down to 1 to 3 colony-forming units per milliliter of blood. It's expensive tech, but for a critically ill septic patient in the ICU, speed is life.
Speed is life. That really sums up this whole episode perfectly.
It does. Fungi are slow, so we have to be fast.
Let's bring it all together in the outro. What is the ultimate takeaway for the listener today? We've covered a huge amount of ground from Murray Chapter 60.
The main takeaway is that fungal diagnosis is a truly multimodal discipline. You cannot rely on just one single test or swab. It is a layered defense. Step 1, the patient. Know the risk factors: neutropenia, HIV, DKA. That's your trigger. Step 2, the stain. Use KOH or Calcofluor for immediate visual clues. And remember, do not grind the mucor. Step 3, the culture. The slow-growing truth. Watch out for the cyclohexamide trap with Cryptococcus. Use the olive oil trick for Malassezia. And step 4, the biomarkers. Galactomannan, CRAG, and BDG. Use them to see the invisible systemic invaders, but know their structural blind spots.
In a world of increasing immunosuppression, the fungi are rising, and the clinical laboratory is the only way to accurately distinguish a harmless background colonization from a lethal invasive infection.
Well said. It's a high-stakes chess match, and the fungi are patient grandmasters. We have to play our moves perfectly.
Before we sign off, I want to leave you with a final thought to mull over. We talked about mold in the cold, yeast in the beast, right? The classic rules. But recently, we've seen a new, terrifying threat emerge globally: Candida auris. It's a yeast, but it behaves more like a multi-drug-resistant hospital bacteria. It spreads person to person, it survives on dry surfaces for once, and it resists many of our best drugs. It doesn't fit the old mycology rules. The chess match is evolving right now. The fungi are learning new moves. Are we ready?
That is the exact question that keeps modern mycologists up at night.
On that slightly chilling note, let's see if you were paying attention. It is time for the rapid recall.
I'll read the clinical scenario, you think of the answer, we'll pause, and then reveal it.
Let's do it.
Prompt 1. A patient with advanced AIDS presents with fever and profound pancytopenia. A bone marrow biopsy is performed. The GEMSA stain reveals macrophages packed tightly with small intracellular oval yeasts. What is the most likely diagnosis?
Loss.
The answer is histoplasmosis, caused by Histoplasma capsulotum.
Remember, intracellular yeasts hiding inside macrophages equals histoplasma. It's the intracellular Hunter stain. Reference Murray Chapter 60, stains, and direct microscopic examination.
Prompt 2. You have a tissue biopsy from a diabetic patient with severe sinusitis and a black eschar on the hard palate. You strongly suspect mucormycosis. You explicitly instruct the lab not to grind the tissue. Why?
Pause.
The answer is grinding destroys the fragile non-septate hyphae.
Mucormycetes have broad, pocky septate hyphae. They act like fragile glass straws. Grinding mechanically kills them, leading to a false negative culture. You must mince the tissue instead. Reference Murray Chapter 60, specimen collection.
Prompt 3. You suspect a patient has cryptococcal meningitis. You order a fungal culture on media containing antibiotics and cyclohexamide. Why is this a dangerous mistake?
Pause.
The answer is Cryptococcus neoformans is highly sensitive to cyclohexamide.
The selective agent designed to kill environmental weeds will actively kill the pathogen you are looking for. You must use paired media, one with and one without cyclohexamide. Reference Murray, Chapter 60, Culture.
Prompt 4. A neonate on total parenteral nutrition develops a central line infection. The blood culture is stubbornly negative on standard media, but the doctor suspects Malassezia furfur. What specific substance must be added to the agar to get it to grow?
Pause.
The answer is olive oil or long-chain fatty acids.
Malassezia is strictly lipophilic. It needs exogenous fat to build its cell wall and survive. Reference Murray, Chapter 60, Culture.
Prompt 5. A severely neutropenic leukemia patient has new pulmonary infiltrates on a CT scan. You order a galactomannan test on the bronchoalveolar lavage fluid. Which specific fungus does this test target? Answer is Aspergillus.
Galactomannan is a major polysaccharide cell wall component shed by Aspergillus species during invasive tissue growth. Reference Murray, Chapter 60, Immunologic Markers.
Prompt 6. You run a 1-formin-3 beta-D-glucan test on a patient with suspected fungal sinusitis and DKA. It comes back completely negative. Does this rule out mucormycosis?
Pause.
The answer is absolutely NO.
Mucormycetes, along with Cryptococcus, typically do not produce detectable beta-D-glucan. They are the major dangerous blind spots of this pan-fungal test. Reference Murray, Chapter 60, Immunologic Markers.
How did you do? If you missed any of those, go back and re-listen to the relevant section. This is high-stakes chess, and you need to know your moves cold.
Until next time, keep your index of suspicion high.
See you on the next deep dive.